Good day, ma'am. Good day, everyone. My name is Kayla Angela Arbabonaria, and our experiment for today focused on mitosis, which is the process of cell divisions that allows organisms to grow and repair damaged tissues. For this lab, we observe onion root tip cells under the microscope to study the different stages of mitosis, such as the prophase, metaphase, anaphase, and telophase. In this presentation, I will explain the procedure we followed, the difficulties we encountered, and what we learned from the lab. So in this lab experiment, our objectives were to observe the stages of mitosis in onion root tip cells under the microscope, to identify and describe the main stages, which are prophase, metaphase, anaphase, and telophase, and lastly, to understand the importance of mitosis in the growth and development of organisms. For our methodology, first we made sure to wear proper lab attire. Next, we collected actively growing onion root tips and carefully cut about 2 to 3 millimeters from the tip, since that's where mitosis happens. Next, we placed the root tip on the slide and added a stain, which is acetylcarmine, and let it sit for about 5 minutes so the cells would take up the stain. Then, we covered it with a cover slip, and we finally observed the slide under the microscope, starting with scanning objective to high power objective, and we recorded all of our observations. The next part of this presentation is about the results and discussions after conducting the experiment or after observing and viewing the specimens, which is the onion root tip under the microscope. Under the scanning objective, the onion root tip appears as a low magnification view showing the general structure of the root. At this power, individual cells and mitotic stages cannot yet be seen clearly. Instead, you can observe the overall shape of the root tip and the stained tissue. The red or pink coloration is due to the stain absorbed by the root, which helps highlight the area where cell division usually happens. This overview helps locate the meristematic region before switching to higher magnifications. Under the low-power objective, the onion root tip becomes more detailed compared to the scanning view. The stained root tissue is clearer and the general arrangement of the cells begins to appear, though individual cells and chromosomes are still not fully distinguishable. At this magnification, the meristematic region can be located more easily, helping prepare for higher power observation. This view guides us to the specific area where mitosis is expected before switching to high power. The cells appear larger and more detailed under the high power objective. Individual cell boundaries becomes clearer, but chromosome structures are still not visible due to poor slide preparation. The area remains mostly in interface with no clear mitotic stages observed. For our conclusion, during this laboratory activity, we were not able to observe the stages of mitosis in the onion root tip cells. This was likely due to several factors, including improper cutting of the root tip, insufficient time to properly prepare the slide, and most cells being in interface rather than actively dividing. Despite this, the activity helped us understand the process of preparing samples from microscopic observations and the challenges involved in studying cell division. It also highlighted the importance of the meristematic region in observing mitosis. Although we were unable to observe mitosis in our experiment, there are some important tips and reminders for next time. When preparing onion root tip slides, it's best to cut the very tip, the meristematic region, because that's where cell division is most active. Making thin and even section helps the cells be easier to see under the microscope. We did it wrong because we honestly weren't prepared, and so we didn't search for or look for on how to do it properly. This It's also important to allow enough time for staining so the chromosomes become visible and avoid rushing. Careful preparation at each step can make a big difference. After staining, gently squashing the sample spreads the cell into a single layer. We also encountered this problem because we spent the most of our time on our first lab experiment on microscopy 2, and we don't have enough time to finish this lab experiment, which is the mitosis, and so we failed to do it properly. Another recommendation is using fresh, actively growing root tips increases the chance of finding dividing cells. Keeping these tips in mind will help improve the results in our future experiments. And that ends our presentation. Thank you.